Hello everyone, my name is Sexton, and the paper that I will be presenting is titled Histone, Hyperacetylation, and Mitosis Prevents Sister Chromatid Separation and Produces Chromosome Segregation. Here are the authors. And essentially this paper is just, as it is depicted in the title, is focusing on keeping histones acetylated and seeing what effect that has on chromosome or sister chromatid separation during mitosis. So to start things off, the cell cycle consists of two phases. You have the mitotic phase and the interphase. And the mitotic phase is the shortest part of the cell cycle, and it takes around 2 hours. And the interphase is between 18 to 20 hours. And in the interphase, it consists of three subphases: the G1 phase, the S phase, and the G2 phase. And so the interphase is pretty much, that is when the cell is replicating its DNA, checking for DNA damage, etc. So mitosis, in mitosis there's 4 stages, and this is the mechanism for cell division. And the 4 stages are prophase, metaphase, and anaphase. In prophase, the chromosomes condense and the microtubules will attach to the kinetochores of the chromosomes. In metaphase, the chromosomes will line up against the metaphase plate, as seen here. In anaphase, these cohesion proteins that are keeping the sister chromatids together, they'll be cleaved, and the sister chromatids will part and go to the opposite sides of the cell, as depicted here. And telophase is the final step in which the nucleus condenses again and the cell divides. So histones, because real briefly we'll talk about histones because they're one of the main points of the paper. They're the structure around which DNA wraps, and they form, that DNA histone formation is what forms the nucleosomes. And so histones are made of 4 subunit proteins. You have the H2A, H2B, H3, and H4. And so H2A, and H2B, they form the protein dimers, 2 copies of those, and then you have the H3 and H4 that forms a tetramer, and those combine to form a histone. So as you can see in this figure, this represents the DNA that is wrapped around the histones, and these are the lysine tails. Now when it comes to talking about acetylation, the acetylation will occur on these lysine tails, and that will help unwound in a way the DNA, so the DNA can be expressed. And this is exactly what they're trying to keep in the paper. They're trying to keep these lysine tails acetylated to see the effect on mitosis. So in the paper they also use a Western blot, and prior to a Western blot you usually do an SDS page, which will get the proteins separated based on their sizes in a gel, and that gel will be sandwiched between 2 different charges. And what you'll run a current through the gel, and you'll incubate it with a primary antibody for the protein, and then you'll wash it to get rid of any of that primary antibody that's left over or unbound. And then you'll treat with the second antibody that will bind to the first antibody, and then you'll add a substrate, and usually there's an enzyme involved in this process that will cleave the substrate, and that will give off a signal that can be visualized using chemiluminescence. So before they began their research, some recent discoveries based on this subject were hyperacetylated histones displayed altered chromatin conformation. Deacetylation before mitosis produced defective chromosome condensation, and presence of hyperacetylated chromatin during mitosis impairs proper chromosome condensation during the pre-anaphase stages. So, and the purpose of this section of the paper that I'm going to talk about is to investigate the role of histone deacetylation on the mitotic process by inhibiting histone deacetylases shortly before mitosis in human primary fibroblasts. And they also use HeLa cells as well. So, as I said, they'll be using human lung diploid fibroblasts, which are MRC5 cells, and they'll also be using HeLa cells. And they'll run antibodies with anti-serine 10, phosphorase 3, and anti-lysine 9 acetyl H3. So, and they'll visualize these specimens using Olympus Vanox microscope, and as mentioned earlier, they will also be using Western blot to test for concentrations. So in the first part of this experiment, they wanted to see what concentration of TSA they would need to use to properly inhibit deacetylases. So TSA, as I said, inhibits mammalian histone deacetylases. And so what they found out is that they only needed 500 nanograms per milliliter TSA to properly acetylate the to keep the deacetylases from functioning. As you can see in this graph, when they used 1,000 nanograms per milliliter, they had the same amount of inhibition as they did when they used 500 nanograms per milliliter. So in this figure, the acetylated form of H3 went undetected in the control, and that is without TSA. And when they added TSA, they could see that the histones remained acetylated, as due with the intense staining that was observed when it was treated with TSA. And finally, they ran a Western blot, which showed an accumulation of acetylated H3 histones with the treatment of the TSA. So next, you want to see acetylation of lysine 9 interference with serine 10 phosphorylation. So once again, they used human fibroblasts, and so the phosphorylated H3 was visible in the metaphase and anaphase part of the mitosis, and they treated these cells with TSA and showed a decreased antibody reactivity to phosphorylation and a decreased H3 phosphorylation when hyperacetylated. So basically what this is saying is that when they treated with TSA, there was a decrease in the phosphorylation of the histones when the histones were when they were kept acetylated. So with this information, they wanted to see, is this possibly due to like an overall protein kinase activity, which is what they viewed next. So in this scenario, they used double immunofluorescence staining with anti-phosphorase 3 antibody and NPM2 antibody. And these antibodies recognize phosphoepitopes on mitotic proteins. And the NPM2 reactive epitopes were unaffected by TSA treatment, as you can see in this figure. As you see with the treatment of TSA, as you see here, there's a decrease in the phosphorylation, but there is no decrease in the activity of the all-around protein kinase activity. So this indicates that there is no general inhibition of kinase activity. So based off this, they're thinking, well, maybe this TSA or keeping these histones acetylated may have an effect on, but not the kinase activity, but maybe the phosphatase activity, which is the next portion that they wanted to look at. So in this section, they used gamma isoform of the PP1 phosphatases to mitotic chromosomes, and they used an antibody against these PP1 gamma phosphatases, and it displayed homogeneous nuclear distribution, and the strong signal was evident in interphase nuclei treated with the TSA, and there was an increased activity of phosphatases. So if you look at the figure here, when there's no TSA treatment, there is no increased signal received from these PP1 antibodies. But when they treated with TSA, it shows that there is an increased activity of the phosphatases, which would probably be correlated with the reason why serine 10-3 or serine phosphorylation was inhibited or decreased. So in this next portion, they wanted to test the concentrations. So they tested phosphorylation, the state, and they used a Western blot analysis, and they investigated H3 phosphorylation in a mitotic cell population. And in this scenario, they used HeLa cells, and they used a thymidine afidoculin block, and they looked at the intracellular levels of phosphorylated H3 histone, did not show significant variations. So you see here throughout the levels, there is no significant variation, as in there was an increase in the intracellular levels of phosphorylated H3 histone. So the concentrations basically were maintained throughout this portion of the experiment. So in conclusion, they found that lysine 9 acetylation does not interfere with serine 10 phosphorylation. And this is probably due to accessibility of entails to antibody recognition. They also discovered lysine 9 acetylated serine 10 phosphorylated tails modify DNA histone interaction. And reduced chromatin accessibility may increase loading on DNA of the chromatin-associated kinases and phosphatases that regulate H3 phosphorylation. And cells entering mitosis with hyperacetylated histones display altered chromatin structure. So lastly, what's next for this paper? Maybe to look at further investigate cohesion factors between chromatids and see what happens there, because in this paper I also talked about chromosome lagging when the cells split, and maybe look at also methylation roles instead of acetylation roles. These are my references.